Okay, thank you so very much for coming to our lesson today. Today we are going to look into cellular respiration. What is that exactly? This is using oxygen in the cells in order to produce energy. How we use oxygen? Oxygen is diffused through the blood vessels. It will be diffused through the cell membrane. It will reach the mitochondrions. These are organelles that exist in the cells, and then it will diffuse into the mitochondria. This is oxygen. Once oxygen is inside, this is going to be taken or used by a complex of enzymes that exist within the internal membrane of mitochondria. And then we will see a cascade of enzymatic reactions that will end up making ATP. ATP is the storage of energy in our body. ATP is adenosine tri or 3-phosphates, ATP. It has 3 phosphates, and those phosphates are bound to each other. It means that you have one molecule of adenosine, and then you have one molecule of phosphate. Then linked to the first phosphate, you have the second phosphate. Linked to the second phosphate, you have a third molecule of phosphate. So now when you are looking into the bond between those phosphates, you will see that when those bonds are broken due to any enzymatic activity like by enzymes called phosphatase or phosphatases, we will see the release of energy. So each time that one phosphate is being released from the ATP, the ATP becomes a DP, so adenosine di or diphosphate, and then the second phosphate can also be released by a phosphatase in order to give us adenosine monophosphate, and then even that phosphate can be also released under the action of another phosphatase for giving us adenosine. Now, each time that one phosphate is released, we have a release of about 7 joule for each mole of the ATP, and that heat is going to allow enzymatic reactions in your cells. So I repeat again, the respiration is the use of oxygen in order to produce ATP. But something I didn't tell you is that we can also produce ATP without oxygen. So respiration is mainly the production of ATP within the mitochondrions with or without oxygen. Now let me go over the content, or I would say the whatever the extract of the book chapter. So the chapter, of course, is cellular respiration. Now, what you are going to learn, you are going to learn to use the appropriate terminology, including glycolysis, Krebs cycle, electron transport chain, oxidative phosphorylation, substrate level phosphorylation, alcohol fermentation, lactic acid fermentation, and ATP synthase. For each of these topics that I showed here, I also added a movie from YouTube, but these are, I checked them all. They are of good quality for the content. They are all available under the link to this document on the course website. So you can always click on those 3, 4, 10 minutes maximum movies, and you're going to be able to look into the graphics as well with good narratives. And of course, you learn much more in maybe 10 minutes with a good movie than 3 or 4 hours of lectures, because you can view, you can of course listen here and look into the again those good graphics. And then explain the chemical changes and energy conversions associated with the processes of aerobic and anaerobic cellular respiration. What is aerobic and what is anaerobic? Aerobic has the word air inside. It means with oxygen. Anaerobic, it means no air inside. And this means that there is no oxygen when we have that type of cellular respiration. So in your cells, all of your cells, living cells, they are all having organelles that we call mitochondrions. Those mitochondrions, they all have not one, but two membranes. They have an internal and an external membrane. The internal membrane of all of your mitochondrions has several enzymes. Each enzyme is having one specific substrate to convert to a product. And now we can talk about a chain of reactions because you have, or we all have, in the internal membrane of mitochondrions, we do have several complexes or several enzymes that work all together. They are making a type of system. And we can talk about respiratory systems in the mitochondria. And when we look into an electron, we see that an electron, and we're going to see how, is going to reach the first enzyme. Then it will go over several amino acids, making that protein for activating that enzyme or a protein in order to convert the substrate to a product. So enzyme 1 of the complex is going to make B from A. Now the electron that is now moving or jumping, I would say, from one enzyme to another one is going to jump to the second enzyme where it will now provoke some changes in the configuration of that protein by changing the configuration of the polar amino acids in enzyme 2. The same thing applies to enzyme 3, to enzyme 4. So you are going to have a chain or cascade of enzymes in the inner membrane of the mitochondrion, where a substrate A becomes B, becomes C, becomes D, and while the substance D is produced, we are also having enough energy in order to produce ATP at the same time that the products are being made. Very interesting so far. I just stop here because we need to now go back a little bit, and then I'm going to show you some, again, some pictures here. But again, after this course or this session, please kindly take the time to watch those movies because it's still fresh in your mind, and you are going really to learn a lot just by watching those movies as well. Now, these are just the I would say activity and thought questions. This is just in class. We are not in class, but usually when a teacher is teaching this material, this topic, they ask students, okay, do you have any fatigue? Have you experienced any fatigue? And students, they say yes. The real thing is that yes, we do have a fatigue. That's because we do not have enough energy in order to produce any movement, for example, all the time. So the ATP has a reserve, or it is stocked in the by the again the mitochondrions in the cells. But when you are using more and more of energy, so ATP, it's obvious that we are going to have again less reserve of ATP, and then we are going to feel that we have a fatigue or we are tired. This is what it says. This is why in the muscular cells we do have a much more, a higher number of mitochondrions compared with another cell in our body. So cells, they can become specialized based on their activities, and then we can have less or more mitochondrions in any cell. And when we have mitochondrions, we are talking, one of the jobs of mitochondrion is to produce ATP. Here, we are just talking about aerobic and anaerobic one. However, just know that when you are eating glucose, when you are eating proteins that become amino acids, and when you are going to eat fats that will become glycerol, all of these glycerol, amino acids, and glucose, they are all going to be used by your mitochondrions in order to produce ATP. Now, the complex of the enzymes that will grab glycerol, for example, in order to make ATP is different from another complex of enzymes that will take an amino acid for making ATP. And it's also different from another, again, complex or system of enzymes that usually get glycerol in order to make ATP. This is why we are going to look into Krebs cycle. Krebs cycle is because we are going to use glycerol, we are going to use glucose in order to produce at the end of the Krebs cycle ATP. However, it's not in this program, in the book I'm obviously here, but when you're going to university and when you are going to learn about biochemistry, this is where you will see that mitochondrion, all of them, they have in their inner membranes enough enzymes in order to produce ATP using different substrates. And those substrates are amino acids or, again, fat or glycerol or glucose. Perfect. And of course, not just glucose, there are so many others, but again, the course is not biochemistry, but I just want you to understand that your mitochondrions are making ATP. These are your power plants, and they use different gas, I would say. They have two types of gas. One of them is glycerol, one of them is amino acids, 20 amino acids, one of them is glucose. Then let's go over the cellular respiration. So here, this is just the chemistry. So here, in this topic, they are just talking about glucose. Okay, perfect. But we know that we also have other systems in the mitochondrion that will use other, again, other substrate as well for producing ATP. Now, when we look into one molecule of glucose, we see that we need 6 molecules of O2. So this is the respiration. We need oxygen. And then we are going to see 6 CO2. So when you are using oxygen with glucose, you are going to produce ATP, and your cells or your mitochondrion is also producing CO2. So that CO2 is going to diffuse out of your mitochondrion, diffusing out of your cell. It will find itself in your blood vessels, where your red blood cells are going to grab those CO2s, and they will bring them close to your lungs, to your alveoli. And from there, we will see that the CO2 is going to diffuse out of the, again, of the blood, and they will find themselves in your lungs. So when you inhale, you are going to get oxygen that goes to your blood through your, again, hemes and red blood cells. They will reach your mitochondrions. Now they are going to allow the production of ATP using glucose. And then what you get is the CO2 that will be produced, and then it will now being exhaled. This is what happens. Now, when we see the production of ATP, we can see the production of CO2 as well as water. So that water, of course, it's going to be diffused out. This is something that is usually, again, in the mitochondrion, in the cell. It's going to be the fluid in your body anyways. Now, what else? Overall, cellular respiration can be broken down into 3 main stages in your mitochondrion. First of all is glycolysis. This is the anaerobic, so no oxygen, anaerobic stage, where a small amount of ATP is produced along with the molecule pyruvate or pyruvate, which moves on to the next part of the process. When we are also lacking enough oxygen when we do exercise, we also have this type of glycolysis, where glucose is being used in order to produce ATP, where we are in a condition where our muscles are not having enough oxygen. This happens usually with people who are running a marathon, for example. And what happens then is that we produce more pyruvate, of course, and from there we may produce more lactic acid. That's also going to cause, again, fatigue as well, unless we are having a resistance training. And in this case, the body is going to adapt itself in order to also use pyruvate in order to produce energy. Then pyruvate oxidation and Krebs cycle. This begins the aerobic, so with oxygen portion where the pyruvate molecule is first altered and then enters a cyclic pathway which generates some ATP along with the high energy molecules of NADH. Look, do not be impressed by these names. Overall, you have a mitochondrion. In the mitochondrion, you have two types of membrane, the outer membrane and the inner membrane. Now, in the inner membrane of your mitochondrions, you do have hundreds of different enzymes. Those enzymes, they can find themselves in complexes where you need to have three, four, five different enzymes to work together in order to get a substrate A and to produce product D, and then to produce now with this cascade to produce ATP. Now it's obvious that we also need some other molecules for the proton transfer, for the electron transfer. Here we talk about FADH or we talk about NADH. These are just different names and different compounds necessary for the production of ATP. Keep it at this level for now. I don't want to confuse you, okay? For now, just stick to the big picture of the mitochondrion, having some enzymes that work in a cascade, getting glucose and producing ATP, and of course CO2. And it also, they use also oxygen. This is all you need to know. Then electron transport chain and chemiosmosis, the high energy NADH molecules are used to generate ATP in a process called oxidative phosphorylation. What happens here is that when we will have a cascade of those changes of different molecules, the glucose is going to give you at the end a compound. However, we will have an adenosine that will be added into a phosphate. Then we will generate enough energy for adding a second phosphate to that AMP or adenosine monophosphate in order to have adenosine diphosphate. Then we have the addition of another phosphate. It will give us adenosine triphosphate or triphosphate. So that ATP is being made by an enzyme that we call ATP synthase. And that ATP synthase needs the molecules that are being made by this enzymatic chain or cascade. So now I repeat again, you have glucose that comes into a chain of, look at this one. This is, for example, these are different. This is the toy of my dog, BB, but it's okay. She's sleeping now. Imagine that this is the mitochondrion. This is the membrane of the mitochondrion. And here I have five different enzymes. Five different enzymes. Now glucose comes here. It will now be transformed by the first enzyme. Then this will be transformed by the second enzyme, by the third enzyme, by the fourth enzyme. And then that product here is going to be recycled. However, anything else that is being produced after these cascades, this is going to be CO2, CO2 that will get out, CO2 that will get out, NADH that will get out here, FADH that is going to get out here. Now, each of these compounds are going to have a role. One of them is to stimulate the ATP synthase. ATP synthase is another enzyme here close to this complex. So this complex is using oxygen, then it's going to produce another compound that will stimulate ATP synthase. This ATP synthase is going to use adenosine here, and then it will produce ATP, adenosine, ATP, adenosine, ATP. So this is the complex. There are several of these complexes, of course, in or on your internal membrane of the mitochondria. This is it. If you know, you can go home now. Thank you so much. I'm kidding. So now let's go over again more information about that. This is the mitochondrion. So this is the external layer, okay? And inside you have the internal layer. Sorry, this is the cell, but this is the internal membrane. So inside, what you can see, let me see if I can make it bigger. Yes, I can. So here you can see that you have the Krebs cycle. In the Krebs cycle, you do have several enzymes. So these are the enzymes that I showed you. And here you have several enzymes. Each time that you are having one enzyme activated, you will see another molecule that is being produced. So what they do exactly? Oh, look at that. We have glucose in step number 1. Then glucose is going to give us pyruvate. This is allowing us to have 1 ATP. Perfect. But then this is not it. Pyruvate now goes into the mitochondrion, and this is now going to undergo some transformation under the action of several enzymes in the inner membrane of the mitochondrion. So pyruvate is going to give us a compound. It's called acetyl-CoA, okay, compound A. It also makes NADH, I would say compound B. Now A goes to the Krebs cycle. It will be transformed in a cascade of enzymatic reactions. This cascade gives us 1 ATP here, and it also gives us several molecules that we call NADH or molecule B. So we have molecule B, 1B here, 1B, 2, 3, 4 molecules of B. We also have 1 molecule of FADH. All of these molecules are going to be found in the inner membrane of the mitochondrion, where they will all stimulate an enzyme that we call ATP synthase. So now you understand that from glucose, we are going to produce several molecules that will all be able to stimulate ATP synthase. And ATP synthase is going to produce ATP. This is all you need to know so far. And again, we are just talking about the Krebs cycle in the mitochondrion. We have other cycles in the mitochondrion as well. We don't cover that in this topic, but for today, we just covered the Krebs cycle for the glucose that allows us to produce ATP. That's it. So now you would like to know, okay, here you can look into again the structure of the mitochondrion. I leave it to you. You can also have different movies that I showed you. We also talk about the glycolysis. You can get more into depth for the glycolysis as well. And here you see that with 1 molecule of glucose, we are going to have at the end, it's not here, I'm going to show you here. Where is that one? It's 36 molecules of ATP anyway. So we'll go over that. Now that was for the glucose. Okay. So 1 glucose after the glycolysis can give us pyruvate. And the pyruvate molecules will go into the mitochondrion, and they are now going to be used in order to produce NADH and FADH. And they will both stimulate ATP synthase, which is another enzyme that will produce ATP. So it gets an adenosine, it will add phosphates into that. When you have ATP triphosphate, it will be released. Another adenosine, 1 phosphate, 2 phosphates, 3 phosphates, released. Another adenosine, 1 phosphate, 2 phosphates, 3 phosphates, released. That's how that ATP synthase is going to work. And again, there's also one movie that you can find from the website where, again, I added the link to that ATP synthase enzyme. Now, we can also look into pyruvate oxidation and Krebs cycle. This is where pyruvate that goes into the mitochondrion is going to, sorry, get into Krebs cycle. How? Pyruvate is going to give us a molecule of acetyl coenzyme A or acetyl-CoA, and acetyl-CoA is going to be used in the Krebs cycle. So within the Krebs cycle, again, you can read that, of course, just to go into more into detail, but within the Krebs cycle, this is the Krebs cycle, where one molecule of pyruvate is going to undergo some transformations. Each time here, you have different enzymes, different numbers, enzyme A, enzyme B or 2, enzyme number 3, enzyme number 4. You do have different enzymes that are all located in the inner membrane of the mitochondria. So now the pyruvate coming from glucose, from glycolysis, comes into the mitochondrion, and now it's going to give acetyl-CoA that will be used. The CoA is going to be released. The acetyl is going to be used. So we are going to have citrate, isocitrate, alpha ketoglutamate, etc., etc. And each time that there is one substrate and one product, we will see the release of one NADH, another NADH, another NADH, FADH here, another NADH, or some ATP. So at the end of the day, these NADHs and FADH2s, they will all go to the ATP synthase. They will all stimulate ATP synthase for producing more ATP. So the overall, I would say, counting, if you would like to do the counting, we will see that overall, okay, this is just the schema to show you that this is the inner membrane. And here you have the glucose. You are going to have the Krebs cycle. All of these are going to stimulate different enzymes. And then your ATP synthase on the bottom right side of this, again, this picture is going to produce ATP. So at the very end, we will see that if we are having, again, the glucose, one molecule of glucose, after the Krebs cycle, we will be able to produce, now you see that 2 ATP for converting, again, GTP to ATP. We have HNADH to ECT, so we have 24 ATPs. Overall, we have 36 ATP molecules produced after using one molecule of glucose. And those ATPs are now going to be released to your body, to your different, again, to your cells. And in each cell has its own mitochondrions. Each mitochondrion is going to produce ATP for that cell. Those ATPs are going to be used by different enzymes or other activities of the cell in order to, again, produce some proteins, in order to do some changes, in order to produce some microfilaments, for example, or anything, wherever we need energy from those ATPs as well. And then we also have now the anaerobic pathway. The anaerobic pathway is when we don't have oxygen. So if you go back here, you will see that for glycolysis and for Krebs cycle, some of these enzymes needed oxygen. If we don't have oxygen or not enough oxygen, this cycle is not going to work properly. And this is why we will see that for the anaerobic pathway, we will see that only few ATPs, 2 ATPs are going to be produced. So here we can talk about lactic acid fermentation in muscle cells. In your muscles as well, when you are running, for example, and you don't have enough oxygen reaching your muscles after a few minutes, in this case, your muscles, the mitochondria, are going to use the glucose. However, that glucose is going to give us pyruvate, and then at the end, it will give us lactate or lactic acid in addition to 2 ATPs only. This is why we see the muscle fatigue. Now, not a similar but not identical reaction exists also in the yeast, where we give glucose to the yeast. They will make it, after glycolysis, make it to pyruvates, and in the absence of oxygen. And then the pyruvate is going to give us 2 ATPs plus 2 molecules of ethanol. which is the alcohol that is, again, that is safe to drink. So this is, again, for showing you that, what happens in mitochondria will depend on the absence or presence of oxygen. In the presence of oxygen, we have 36 ATPs produced, molecules of ATP based on use of one molecule of glucose. If this is a fermentation in the, again, in the muscle, we call that a lactic acid fermentation. This is where one molecule of glucose gives us only 2 ATPs. It gives us still energy, but not as much as with oxygen. It also produces lactic acid in the muscles. And of course, it's going to provoke the fatigue aspect. If not, in the yeast, we see the alcohol fermentation. Perfect. This is just for, last one is just for talking about the Krebs cycle and the Calvin cycle. So on the left, we are looking into, again, the production of ATP in plant cells. That's what we saw last week. Today, we are looking into the Krebs cycle and what happens in the mitochondria. Again, these are very similar but not identical processes. This is the way that nature uses glucose in order to produce ATP. But again, it's not just using glucose for making ATP. The nature, our nature allows us also to burn or to use fats and amino acids for making ATP as well. This is something that that is not covered in this year's class, of course, but when you're going to go to university and you're going to learn more and having biochemistry classes, you're also going to look into the other different types of molecular transformations that we can see in the mitochondria. So my friends, I just finished with, again, today's lecture. So I just want to thank you so much for, again, your attendance. Thank you so much.